Hi. In this research project, we are going to investigate the effects of 3 different mouthwashes on salivary microbes. So I have here 3 commonly used mouthwashes purchased in trial size. I have Listerine, I have Scope, or what used to be Scope, that has alcohol in it, and then I have Crest without alcohol in it, or an alcohol-free mouthwash. So the way that this particular lab is going to work is I'm going to collect some saliva. I'm going to collect at least 2 mils in here. I will not be filming that part so that you can keep your lunch down while I'm bringing mine up. So you'll just have to trust me that yes, I'm going to be spitting in here and there's going to be 2 to 2.5 mils in there. Once I have that saliva, I'm going to separate it out into 4 small microfuge tubes, kind of similar to the ones that we used in Lab 16, the genetic engineering labs. Are pretty small, little small tubes. And then what I'm going to do is I'm going to be adding some of the mouthwash to each tube. I'm also going to have a control where I have some saliva with just water. That control will allow me to, or allow you, to analyze the number of countable and platable microbes that I started with in my saliva. Now, even though my saliva probably has hundreds of different species of microbes in them, not all of the microbes will be cultured here. So viruses, for instance, won't, and then some of the bacteria won't grow in agar either. We haven't been able to get them to grow in agar. So understand that the number of microbes that you see in the control plates is actually lower than the total number of microbes that would be in my saliva. But we can at least investigate, well, what is the concentration of colony-forming units in the saliva, and then how does Crest, Scope, or Listerine reduce that? What, maybe what percentage of those microbes are reduced by these mouthwashes? So in order to do that, I need to make sure that I mix my saliva with the mouthwash for the prescribed time. And when I'm looking at the instructions on these, the prescribed time looks to be about 30 seconds of gargling. So to most accurately mimic that, the way that I'm going to do this experiment is I'm going to use half a milliliter of saliva with half a milliliter of whatever mouthwash is there, or water for the control. And I'm only going to let them sit for 30 seconds before I start my serial dilutions. So I'm going to go collect some spit and then I'll be right back. Okay, so I collected my saliva. Super glad I did not put that on camera. I just had lunch. It was really chunky, and I had cheese. As you can probably see, there's a little bit of color and some floaters in there. But I collected all my saliva into one tube and then split that tube up into 4 of these small tubes. And the reason why I collect all the saliva at once instead of spitting into each tube is because as you collect saliva and spit, and then you try to get more saliva to get more spit, it's possible that you actually decrease the number of microbes in subsequent spits, simply because you've gotten most of the microbes out in the first batch, loogie, expectorant, I don't know, whatever you want to call it, the first batch of saliva versus subsequent batches. So to make sure that the numbers of microbes are pretty consistent for all the tubes, I collect all my saliva into one tube, mix it up, and then aliquot it out into these little tubes. Now the control is going to be run just like the ones that we do with the, or just like the experiments that we do with the mouthwash, but instead of adding 500 mils of mouthwash, I'm going to add 500 mils of water. But just like the mouthwash procedures, I need to make sure that it only sits for 30 seconds mixed before immediately starting my serial dilutions. Because I'm working from these small tubes, I'm actually going to use a micro pipetter rather than a full pipette to do my first set of dilutions, just because it's a little easier for me to work with these two put together rather than the big unwieldy pipette that you would normally use. I will be using those in a moment. Okay, so let's go ahead and get started. I'm going to add half a milliliter, maybe, there we go. So I pulled out 500 microliters of water, and I'm going to mix it with my control and then time it for 30 seconds. Okay. Okay, so it's been 30 seconds. Let me take out 1 ml and go ahead and dump it into bottle A. As shown here. Okay, now once I've done that, I've moved it into bottle A. I've got time now. That 30 seconds is really just about what's being mixed in the tube before doing your first dilution. The reason is because once it gets diluted into bottle A, whatever is antimicrobial in your original test tube, in our case, water is not antimicrobial, but we expect that these will be antimicrobial. Whatever is antimicrobial about that particular solution, mouthwash, gets diluted out to such an extent in bottle A that it usually ends up stopping whatever antimicrobial reaction was occurring. So now that it's diluted out, I have a little bit more time to play with. So that was my control. That was, again, half a milliliter of water, half a milliliter of saliva. And now what I'm going to do is I'm going to follow Lab 8 procedures to do my set of serial dilutions. So you'll recognize that I have my 3 bottles up here, A, B, and C. And just like Lab 8, I'm going to have 4 Petri dishes. And my dilutions are going to be a 10 to the minus 4, 10 to the minus 5, 10 to the minus 6, and 10 to the minus 7, exactly as prescribed in Lab 8. All right, so I'm going to go ahead and do that. So now that I've done my control, all I need to do now is make pour plates of these. So I'm going to go ahead and stop and then zoom in on the pour plates once the agar is out and cool enough to actually make pour plates. Okay, we're ready to make some pour plates of our control. So I've pulled the agar out of the water bath. It's cool enough that I can now make pour plates. Typically I would get a little bit of help with this, so if you don't have that help, it's going to be a little bit messy. But you're going to do the same thing that you would normally do. Pour it in and then mix it with the water droplets. Get a good swirl, a good mix. Okay, then we're going to let those plates harden, and then we're going to put them in the 37-degree incubator overnight and look at them in the morning. So I've already done the serial dilutions for the control, which is water. And now what I'm going to do is I'm going to do the same experiment, but for scope. Now I've already done something very similar for crust and for Listerine. So I'm going to show you scope because all of the mouthwashes follow this procedure. Now what's different about this particular adaptation to the serial dilutions is that I expect that this mouthwash will actually kill off a lot of the microbes in my mouth. So therefore, if I follow standard Lab 8 protocol and do serial dilutions to the 10 to the minus 4, 10 to the minus 5, 10 to the minus 6, and 10 to the minus 7, I expect to get almost no colonies, certainly nothing within 30 to 300 colonies, which is what a countable plate is. So therefore, I'm going to scale my serial dilutions down and make them more concentrated. So I'm going to use just two bottles, a bottle A and a bottle B, like so. I'm going to take my mixture of scope and saliva after it's sat for 30 seconds, and I'm going to pipette 1 ml into bottle A. And then I'm going to take 1 ml from bottle A and actually put it on a 10 to the minus 2 plate. I'll take 0.1 ml from bottle A, and that will be a 10 to the minus 3 dilution. I'll take 1 ml from bottle A to bottle B, and then again 1 ml from bottle B to make a 10 to the minus 4. So notice that I'm skipping 10 to the minus 5, minus 6, and minus 7 because I don't expect those to have colony numbers between 30 and 300. All right, so let me get started. I'm going to start by taking a half a ml of scope and mixing it with my saliva and then letting it sit for 30 seconds. Okay. So now I'm going to take 1 ml from this solution and mix it into bottle A. Chunky. All right, we'll go ahead and mix bottle A really well. And now I have a little bit of time because I've done my dilution, my first dilution, which stops the reaction of whatever was in scope that was killing the microbes. So now I have some time to make sure that I do my serial dilutions correctly and accurately. So with my first pipette, I'm going to take 1 ml from bottle A and place it on the 10 to the minus 2 plate. I'm going to take 0.1 ml, and no, that did not work. I'm going to try this again here. Sometimes the airflow with these pipettes doesn't work real well. Okay, let's try this again. I'm going to take 0.1 ml. There we go. And add it to the 10 to the minus 3 plate. And then finally, I'm going to draw up 1 more ml maybe. The airflow is, I don't have a complete seal on this one. Okay, let's show this again. Okay, better. And add it to bottle B. Mix bottle B. And take 0.1 mil, or sorry, 1 full mil from bottle B and put it into the 10 to the minus 4 plate. Now after I do that, all I need to do is add the BHI agar to make the pour plates and put them in the incubator and check the results tomorrow. Hey, so it's the next day, and we are looking at the results of our pour plates. And so I have unstacked the pour plates, and I've started to take a look at them. And so what I'm going to do now is I'm going to actually just take photos of the pour plates, and I will go ahead and embed them into this video. You are now going to be presented with the results of the pour plates. The first set will be saliva and water, so this was the control. It will always start with a picture of all of the pour plates from that set, and then it will transition to a video where you start to see that I bring the plate forward and use a Sharpie to mark where the colonies are. So you need to follow along with the Sharpie in terms of how many colonies there are. If there are too many colonies to count, I will simply write TNTC, too numerous to count. Good luck.